We can hear you.
Awesome. So we're going to be offering these sessions each Tuesday at 4 o'clock. They'll last from 45 minutes to an hour. That's the plan. I would like, in a perfect world, for everyone to mute your microphone when you log in and turn on your camera if you have one. As we're going, feel free to unmute and ask questions. We want you to ask questions if you have them. I'm operating this alone, so if you have a question, it will be best if you just unmute and ask it. Typically, I would have someone to help look at the chat, but I don't have anybody to do that today, so I may not see your chat questions. So just ask if you have questions. Again, we are going to do these each Monday at 4. The same Zoom link that you use today will work for all of the sessions. We are recording these sessions, and we're going to post them up to our YouTube channel so that people who couldn't be here live can access them. So those will be available, and I'll send direct links to your teachers. But our YouTube channel is A+ College Ready Science. You've got to spell out plus, A+ College Ready Science. Each week, we're going to look at a different unit. We're going to hit some of the highlights from each unit. This unit, Unit 1, we're just going to go in order. And I'm going to look at some what should be, I hope, easy points for you from this unit. So bear with me. I want to get to a certain slide here. And I want to talk to you a little bit about experimental design. There's always questions about setting up experiments on the AP exam. Let me find the right slide here. I'm in the wrong place. There you go. And this, I hope, should be straightforward and easy, but it's amazing how many times people miss this on the AP exam. So one of the things that people have a really hard time differentiating between is, number one, the control group and the experimental group. So anytime you do a scientific experiment, you're going to take all your subjects and you're going to randomly divide them into at least 2 groups. You want the groups to be the same. You want if you're testing a drug, you want the people in the 2 groups to be about the same average age. You want the same amount of males and females in each group. You want them to have the same kind of health status. You want essentially everything to be the same except the one thing that you're testing. In my example here, it would be the drug. That's important. The thing that you're testing, you're going to expose individuals in the experimental group to that thing. And the thing that you're testing is not going to be given to the individuals in the negative control group. And we're going to talk briefly about 2 kinds of control groups, negative and positive. By far, the one you're going to deal with most often in AP Biology is a negative control group. This is essentially one that is not treated with the independent variable. So let's talk about 2 things in an experiment. The independent variable is typically the thing that you're testing. For example, if you wanted to know the effect of fertilizer on plant growth, the independent variable would be the amount of fertilizer, or if you gave them fertilizer at all. On the other hand, the dependent variable is the thing that changes because of the independent variable. So again, if we were looking at the effect of fertilizer on plant, let's say plant height would be the dependent variable because it is changing because of the independent variable. When you're graphing these variables in any kind of graph, whether it's a line graph or a bar chart, the independent variable is going to get graphed on the x-axis, and the dependent variable is going to get graphed on the y-axis. So again, a negative control group is one type of control group. It's a control group that's just not exposed to the independent variable. So in that fertilizer group that I just talked about a minute ago, the fertilizer study, again, you're looking at the effect of fertilizer on plant height. The negative control group would be a would be a group of individuals that didn't get any fertilizer at all. That's a negative control group. Now, on the other hand, you might have positive control groups. These kinds of control groups are not nearly as common in science, but they're used to get a certain result. For example, maybe you're looking at, again, the fertilizer example with plants. So instead of giving them the fertilizer that you're actually testing in the study, maybe you give them a different brand of fertilizer that you know works, just to show that it's the fertilizer and not the plant that's affecting the outcome. But by far, the most important kind of control group you guys need to worry about is a negative control group. It is one that is not exposed to the independent variable. If you're doing a drug study or something like that, you're going to give individuals in that control group a placebo, essentially a fake. If you're giving tablets, they're going to get a fake tablet. It's probably just made of sugar or salt. If they're getting injections, they're going to get an injection of saline, probably just salty water with no active ingredient in it. That being said, let's look at a couple of questions that deal with that. Typically, we're going to try to do these sessions with Pear Deck, which is a software that will allow you guys to be interactive, but I had some tech issues today. I'm not sure what's going on with Pear Deck. I couldn't get it to function the way I wanted it to. So today we're just going to do it with this PowerPoint. I'd like for you guys to read this little blurb up at the top of my screen, and then go ahead and try to answer these three questions. You got questions 1, 2, and 3. So you can either jot them down on a sheet of paper or just in your head, keep up with what you think the right answers are. So again, read the blurb at the top of the screen and then answer the 3 questions down below it. I'm going to give you about 2 minutes to do that. And please make sure your microphones are muted. I can hear a lot of background noise from somebody. All right, so let's look at this. So we've got the group A rabbits, 1,000 male rabbits are tested. They're given, the group A rabbits are given an injection of a drug that's supposed to help control blood pressure. The other rabbits, the group B rabbits, they didn't get anything. They didn't get an injection. They didn't get a pill. They just didn't get anything. All the rabbits are the same breed, fed the same type and amount of food, housed in identical conditions. Those things are called constants. And again, you want everything to be the same between your two groups, except the independent variable, which in this case, the thing we're testing, the independent variable is this drug, this blood pressure drug. And the dependent variable would be the blood pressure. What is the actual blood pressure of these rabbits? We're thinking that the dependent variable changes based on the presence or absence of that drug. So question 1, which group acted as the control? I'm hoping all of you came up with group B. Those group B rabbits were not exposed to the independent variable. That's what a control is, at least a negative control. Now the next question is the important one. Were the control and experimental groups adequately matched? You got to think, we're working here with blood pressure. And we're wanting to essentially keep the blood pressure lower by giving these rabbits this drug. The rabbits in group A were given a shot. I don't know about you, but anytime I'm going to get a shot, or even I think about getting a shot, that's going to cause my blood pressure to go up. The rabbits in group B weren't given anything at all. No injection, no nothing. So at first you might have thought, yeah, that's a good control. They're not getting the drug, but they're also not getting the shot, which is an important part of this experiment. So the answer here is C. For this to have been a good study and for us to expect accurate results, we would have given group A rabbits the injection with the drug in it, and we should have given group B rabbits an injection, but with probably saline or just some non-active ingredient inside the injection. That way we take out the effect of the injection, but we don't take out the effect of the drug. Either it's present or it's not. And in this case, that injection of saline would have been a negative control. The rabbits in that group would have been in the negative control group. They were not exposed to the independent variable. All right, I'm going to go to slideshow mode here for a second. So question 3, assume an adequate control was used. So let's assume that we did give the group B rabbits an injection. Which hypothesis could be tested using this experimental design? Someone want to unmute and tell me what you said on number 3?
Choice A.
Okay, I hear A.
D.
I hear D.
I got D too.
All right, so let's look at A first. So the drug lowers blood pressure in some breeds of rabbits, but not in others. Well, if you go back up to the stem of our question, it doesn't really say anything about the breed, right? Let's see. Is there anything at all about the breed? Yeah, same breed. So they're all the same breed. So that little S right there really makes A be wrong, right? Because we only look at one breed. We don't have any way to know what happens in other breeds. We're only looking at this specific breed of rabbit. So A cannot be the right answer simply because of that. We're only looking at one breed, and we don't know what happens after that. So B can't be right either because we didn't administer the drug orally, so that can't be right. C can't be right because we didn't have any female rabbits in either group, so we don't know about the female rabbits. Really, the only hypothesis that we can test here is, does this drug lower blood pressure in this breed of male rabbits? That's really the only thing we can answer. So D would be our correct answer there. Any questions on this set?
Doesn't say anything about male rabbit.
Yeah, so if you look up at the stem, it says 1,000 male rabbits were tested.
So would number 2 be an example of a positive control group?
No, it's negative. And remember, what makes it a negative control group is that the rabbits aren't exposed to the independent variable. The thing we're testing here is this drug. The rabbits in group A get the drug by injection. The rabbits in group B are going to get an injection, assuming that we do it the right way, but there's not going to be any of the drug in it. So they're not exposed to the drug at all. That's why it's a negative control group. Does that make sense?
All right.
All right, so let's go a little bit further with this. So this is an actual free response question off an AP exam about 4 or 5 years ago. Same format that we use today. This is a free response question that I'm hoping that you guys would get all the points on. So take about 2 minutes and just read through everything you see here on the screen and think about how you would answer this. If you have some paper, you could sketch an answer down on paper if you have something there in front of you. All right. Again, I'm hoping that you think this is a pretty straightforward question. So we've got these seagrass. They're aquatic plants. They reproduce sexually, like most plants do. The males make pollen. Typically, the pollen is carried just by moving water to the females, where it's going to fertilize the eggs in those female flowers, produce seeds, produce fruits, ultimately produce another plant. That's typically how plant reproduction works. But the researcher in this study claims that mobile just means moving, aquatic invertebrates, little insects or shrimp or something like that in the water can transfer the pollen from male to female. So that's his hypothesis. The aquatic invertebrates can move the pollen from male flowers to female flowers, even when there's no movement of the water, the absence of circulating water. So to make this investigation, he's setting up some aquariums to see what's going on. And if you look at part A, use the symbols below and the template aquariums to demonstrate the experimental design for testing the researcher's claim that mobile aquatic invertebrates can pollinate seagrass in the absence of circulating water. Draw the appropriate symbols in the negative control aquarium and the experimental aquarium. All right, so notice down at the bottom, we've got two aquaria. And remember, what we're trying to test, you got to always keep this in mind. You're trying to test, can invertebrates move pollen from males to females? So in the experimental group, you've got to have a male flower. So you would draw your little rectangle. You got to have a female flower. And then what we're trying to test is, can the invertebrates move the pollen back and forth? So they've got to be there too. Now, in reality, you would have probably multiple of each one of these things, right, in a real test. But this question specifically says, draw the appropriate symbols. Do not use any symbol more than once in the same aquarium. So what we're showing in the experimental group is we've got male flowers, we've got female flowers, and we've got these little invertebrates. Now, on the other hand, we've got the negative control group. Someone unmute and answer this question for me. We said for it to be a negative control group, what's missing? What does it mean to be negative control group?
The negative control aquarium will be missing an invertebrate.
Right, which is what part of an experiment?
That is the
Yeah, in this case, the invertebrate is the independent variable, right? That's what we're testing. We're wanting to know, does that invertebrate affect the pollination rates? So for it to be a negative control group, the only difference between the negative control group and the experimental group is the absence of the independent variable, which means in this aquarium, we ought to show male flowers. We ought to show female flowers. The only thing that we're not going to show are little invertebrates. Simply for doing that, you got the points, all of the points for part A. I'm hoping that was pretty straightforward and easy for you. Now on part B, notice identify the independent variable in the experiment. So in just one sentence, you're going to say the independent variable in this experiment is, and you guys would say again, what's the independent variable?
The invertebrates.
I would say the presence of the invertebrates. You probably would get the point just for saying the invertebrates.
I would
say the independent variable is the presence of invertebrates. Period. You got points for that. And then the other part, predict the experimental results that would support the researcher's claim that mobile aquatic environments can transfer pollen from male to female flowers in the absence of circulating water. So remember, in each of these aquaria, the water is still. There's no circulating water in either one. And we want to know what results would show us that it's the invertebrates here that are moving the pollen. Well, what would we expect to see in the negative control group? What would we expect to find a few days, few weeks later? In the negative control group, what kind of results would we expect?
That they didn't reproduce.
Yeah, they didn't reproduce. So there's no pollinated flowers, which would mean there's no seeds, no fruits, no babies. Okay? So however you want to say that, there's no pollination in the negative control group. We're expecting pollination in the experimental group. Now, realistically, might there be a little bit of pollination in the negative control group? Because that pollen's going to float, right? Even if the water's not circulating, there's still going to be some floating pollen in the water that may make it. So it might be better to say that we expect to see much greater rates of pollination in the experimental group than we see in the negative control group. That would probably be a better way of answering this question. Does that make sense? So what I'm hoping that you guys see is that the questions like this should be straightforward and easy. There are lots of them that are this easy on the exam. Sometimes they'll be in a free response question like this. Sometimes this same kind of thing will be asked in a multiple choice question. But you've got to be able to know the difference between an independent variable and a dependent variable. You've got to know the difference between a negative control group and an experimental group. And if you know those things, you can answer essentially any of these questions. Any questions on this before we move on to the next point? All right, we'll get a little bit more complicated on the next part. We're going to talk about what's called hypothesis testing. I know there's a lot of words on the screen here. You don't have to try to read all this. I'll kind of talk you through what you need to know. So it's almost impossible to prove a hypothesis completely true. So instead, what we're going to do in what we call statistical hypothesis testing is we're going to come up with two hypotheses. One of them is called the null hypothesis. And I'm hoping you guys know the word null. What does it mean? Just in regular, general English language usage. What's null mean?
Error, nothing.
Say again?
Not valid.
Okay, it can be not valid. It can be none or no. I want you to think of it that way, like none or no. Because what the null hypothesis is, it's essentially a statement that you're going to see abbreviated as H naught. That's like H with a 0 as a subscript. It's essentially saying that there's no difference, no statistically significant difference between the groups that you're comparing. So this could be between your control group and your experimental group, or it could be between a sample that you've taken from some huge group and the whole population. The null hypothesis is essentially there's no difference between your two groups, the groups you're comparing. Another way of saying it is that your treatment, you could also state it as your treatment has no effect on the individuals. So if we go on down and look at the paragraph right here, let's look down at the bottom for the example. So here it says, for example, if you're testing the idea that sugar makes kids hyperactive, your null hypothesis might be there's no difference in the amount of time kids previously given a sugary drink and kids previously given a sugar substitute drink are able to sit still. So you've got these two groups of kids. The kids in the experimental group, they get a sugar drink. The kids in the negative control group, they get a drink without any sugar. So they're not exposed to the independent variable. And your null hypothesis here is that there's no difference between their activity levels. On the other hand, you also typically come up with what's called the alternative hypothesis. And this is abbreviated as H subscript A, HA. This is what you really think is happening. So in this particular experiment that I described above, my alternative hypothesis might be that sugar causes kids to be more hyperactive, or sugar causes kids not to be able to sit still as long. Sugar causes kids to move more. That's my alternative hypothesis. It's hard to test the alternative hypothesis, but what we're going to do is we're going to test the null hypothesis. And if we prove that there is a statistically significant difference between our two groups, then we're sort of sideways proving that sugar caused the kids to move. So by disproving the null hypothesis, you're giving some evidence to back up to support the alternative hypothesis. And let me show you what I mean there by looking at some examples. Here I say that same kind of thing a different way. So it's important to realize that hypothesis testing does not allow proof or even acceptance of an alternative to the null hypothesis. Typically, it comes down to whether or not there's enough evidence to do what we call rejecting the null hypothesis. If we reject the null hypothesis, we're rejecting the idea that there's no difference between our two groups, which means there is a difference. And if there is a difference, that's evidence for that other hypothesis that we came up with, the alternative one. All right, so we're going to do several kinds of statistical tests, but the one we're going to focus on today is called the chi-square test. Some of you unmute and just give me a yes if you've done some chi-square problems before.
Yep.
Yes.
Yes.
Yep.
Yes.
Yep.
Yes.
Any no's? All right, so it sounds like most of you have seen chi-square before. This formula is on your AP Biology formula sheet, which you'll be able to use throughout the exam, so you don't have to memorize the formula. It's a test that's essentially used to compare two groups of data, usually categories, where you're either counting the individuals in the two categories and just comparing there's 10 of these and 20 of those, or sometimes it could be percentages. 20% are in this group and 80% are in this group. So you're either comparing counts of categories or percentages of categories. Chi-square is not used to compare averages. There's another test that we'll sometimes use to do that, and that's either the t-test or the ANOVA test, which we're not going to deal with today. We're going to deal with chi-square. It's used to compare two groups of either counts or averages. We're counts or percentages, I meant to say, between the two groups. So I'm going to show you the basic way to do this first, and then we'll come back and we'll look at some of all these words in a minute. Let's go to an example first. All right, so this is the first question we're going to do. So chi-square example 1, the M&M company claims that its bags contain 30% red, 20% brown, 10% yellow, 20% green, and 20% orange candies. And there is a real number. It changes ever so often. So these are just numbers I made up. These are not the real numbers. But let's assume these are the real numbers from the M&M company. So that's the population. If you took all the M&Ms in the world and you counted them and you grouped them, these are the percentages the M&M company says you're going to get. We buy just a bag or two, and we pour them out and we group them. And here's what we get. We get 50 red, 60 brown, 30 yellow, 40 green, and 20 orange. And we want to compare is what we got in our bags, does that match up with what the M&M company says we should get? So let's think of this as a sample. And let's think of these numbers as representing the whole population of all the M&Ms in the whole world. And we're trying to compare the sample to the population to see if the numbers match up. So first of all, we've got to state a null hypothesis. Somebody again unmute and tell me what we said null means.
None.
None or not. So our null hypothesis is always going to be there's no statistically significant difference between our sample and our population. And you could write that in a lot of different ways, but you might just say there's no statistically significant difference between the numbers in each color category in our sample compared to the population or compared to what M&M says should be there. Lots of different ways you could write it, but that's essentially what you're going to say. So now we're going to actually do the mathematical test. And I'm going to go to a slide here and show you how to do this. I would prefer, if you're doing a chi-square, to do it in a table. And first of all, you need to write out all your phenotypes, which in this case would be, remember, phenotypes means appearances, colors in this case. So we've got red, brown, yellow, green, orange. So let's write those in. Red, brown, yellow, green, and orange. All right, when we're dealing with chi-square, somebody tell me what the O stands for when we're dealing with chi-square.
Observed.
Observed.
Observed. So that's when you do an experiment, it's what you actually got when you did the experiment, right? So in this case, our experiment's really just opening up a bag of M&Ms and counting them. The observed would be what we counted. So let's go back and look at those numbers. Here's what we counted. 50 red, 60 brown, 30 yellow. Whoops. Let's see, 40 green and 20 orange. All right, somebody tell me what the E stands for.
Expected.
Expected. What do we expect to get if these numbers were what they were supposed to be? So let's go back, and we've got some percentages here. So 30, 20, 10, 20, 20. Let me write, I'm just going to write these numbers down over here. 30, 20, 10, 20. These are the percentages that the company says we should get. We'll come back to those in a second. To calculate the expected numbers, though, the first thing we need to do is add up all of our total sample. How many individuals are in our sample? So let's go 50 plus 60, 110, plus 30, 140, plus 40, 180, plus 20, 200. So we got 200 total M&Ms. So somebody unmute and tell me how many we should expect to have that are red. What should go in this space right here?
60.
60. And how'd you get that, sir?
I just multiplied 30% times 200.
Yep. So you take the total number of sample individuals, you multiply it by the expected percentage, which is 30%. So 200 times 0.30. So we got 50, but we expected 60. For brown, we got 60, but we expected 200 times 20% times 0.20, which would be 40.
40.
All right. For yellow, we got 30, but we expected 10% of 200. 200 times 0.10, which would be
20.
20. For green, we actually counted 40 green M&Ms, but we expected 20% of 200, which would be 40, right? And for orange, we got 20. We expected, though, 20% of 200. 200 times 0.20, which is 40. Everybody with me on how we got the observed and expected numbers? All right, to finish this thing up, you just do what these column headers tell you to do. So the next column is O minus E, observed minus expected. So in this case, it'll be 50 minus 60 to get negative 10, 60 minus 40 to get 20, 30 minus 20 to get 10, 40 minus 40 to get 0, and 20 minus 40 to get negative 20. All right, then you go O minus E squared. So you square this column. So negative 10 squared is positive 100. Remember, negative times negative is positive. Everything from here on out ought to be a positive number. 20 squared is 400. 10 squared, 100. This is 0. This is 400. All All right, and then the final column, O minus E squared over E. So we need to take the value from this cell and divide it by this cell. So in this case, 100 divided by 60. So that's 1.67. All right. Then this one would be 400 divided by 40, 10. 100 divided by 20, 0 divided by 40, and 400 divided by 40. All right, so we filled out our table. Now we've got to calculate what's called our chi-square statistic. And this is where that formula comes in off of your formula sheet. So chi looks like a fancy X, but that's a Greek letter chi. And remember, it's equal to the summation of O minus E squared over E. So what is that formula essentially telling us to do with our table?
All of our numbers together.
Yeah, it's just saying take this last column and add them up, right? Add up everything in the last column. So when you do that, let's say we got 10 plus 10, 20, plus 5, plus 1.67, you get a chi-square value of 26.67. Now, by itself, that doesn't really tell you much of anything. To use this chi-square statistic, you've got to have something to compare that to. And you're going to compare that number to what we call a critical value. And you get that critical value from a table that's given to you on your AP Biology formula sheet. Here's the table. So you've got to know how to use the table. And before we do, let's go back to our data real quick. So if you look, we had 5 phenotypes, right? 5 colors. Keep that in mind. So to use the table, you first got to know how many degrees of freedom. The degrees of freedom are the number of phenotypes you have minus 1. So we had 5 phenotypes, 5 colors. Our degrees of freedom in this case would be 4. Unless you're told differently on the AP exam, you're always going to use the 0.05% p-value. And essentially what that means, the p-value of 0.05, it means there's essentially a 5% chance that we're going to make the wrong conclusion based on our data, just by chance. There's a 5% chance that we're going to make the wrong conclusion, that the data is just weird like that. It happens. But 5% is sort of the standard that scientists are willing to use. They're willing to take a 5% chance that they're going to make the wrong conclusion. Anyway, using this column and this row, we come over here and we get a critical value of 9.49. That's the important cell for us to think about. So let's go back to our table. So we had a chi-square value of 26.67, and we had a critical value, what was it, 9 point, anybody remember what it was, 9 point what? 9.49. So we have to compare these two numbers. If your chi-square value is greater than your critical value, we're going to reject the null hypothesis. If chi-square is greater than the critical value, you're going to reject your null hypothesis. So let's go back to the question and let's think about what that means. What again was our null hypothesis? What should always be your null hypothesis? Somebody help me out. What do we say null means?
Something including that there's like no statistically significant difference.
Yeah, so there's no difference between this group and this group. Well, our data, because of our chi-square value being bigger than our critical value, we're rejecting the null hypothesis. Which means, what are we saying? If we're rejecting the null, what does that mean?
There is a statistic.
There is a difference. Something's going on. something's weird about these M&Ms. The M&Ms in our bag do not match up with what the company says they ought to be. So by rejecting the null hypothesis, we're essentially supporting an alternative hypothesis. Something weird is going on with these M&Ms. Does that make sense? All right, so let's do a little reviewing of some genetics now while we're reviewing chi-square. So in the P generation, so that stands for parental, the parental generation, a female fruit fly, homozygous for white eyes, is crossed with a wild type male. That means he's homozygous for red eyes. So we've got a female with white eyes. And I'm going to just draw some symbols up here for us real quick. So that's a symbol for female. Just looks kind of like a stick man. It's a stick woman in this case. And she has white eyes. Somebody tell me what homozygous means.
Either
alleles
two
are
dominant
different.
alleles or two recessive alleles.
Yeah, it means they have, for most traits, there's two alleles that control the trait. Homozygous means that in this individual, they're both the same. In this case, this female fruit fly has, for eye color, two alleles. They're both for white. The male, on the other hand, has two alleles. They're both for red. So we're breeding these together. This is our P generation. The F1s are their babies. So these flies have these babies. All the F1 male flies have white eyes, and all of the F1 females have red eyes. How could that be possible? Somebody help me out. How in the world could we started with females with white eyes and males with red eyes, and they have babies, and we end up with the opposite. We end up with white-eyed males and red-eyed females. How could that be possible?
It's a sex-linked trait, so the gene is on the X chromosome.
Yep. So what she's saying is she's saying that the genes for eye color must be on the sex chromosomes, one of the sex chromosomes. In fruit flies, sex chromosomes work exactly the same way they do in us. Females are XX, and males are XY. So I'm going to think about the parental generation here first. But before I do, let's think about one other thing. Which one of these eye color genes must be dominant? White or red?
The wild type.
Say again?
The wild type.
Red.
Okay, so let's think about it this way. So these F1s down here, the males, they end up with white eyes, but the females end up with red eyes. So this male, where did his X chromosome come from? Where does a male get his only X chromosome?
The mom.
The mom. Well, who was the mom? Her. And what are the only alleles she had for eye color? She only had white-eyed alleles, right? So since mom was white, it makes sense that the male would have white eyes if it's X-linked. On the other hand, the female right here, she got one X from mom, but where'd the other X come from?
Dad.
Came from dad. Dad only had what kind of alleles for eye color? Red. Mom only had white, which means that this F1 female got, I'm going to use a big R for red and a little R for white. She got this allele from dad. She got this allele from mom. This guy right here got his allele from mom, and it was recessive and white. What does the Y have to do with this?
The Y doesn't get an R.
Yeah, it doesn't have anything to do with eye color. It just happens to be the other sex chromosome. The genes for eye color are linked to the X chromosome, not the Y. So when we're doing these little subscripts like I did here, you don't put anything on the Y because it doesn't have that gene. Everybody with me that those are the phenotype, the genotypes rather, for our F1 offspring. Everybody understand why that's the case? All right, so if you look at question A, what is the most likely mode of inheritance for the white eye color trait? How is this white eye color trait passed from offspring to offspring? We says it's linked to what?
The X chromosome.
X chromosome, and it's what kind of trait? Dominant or recessive?
I think it's dominant.
Now I'm talking about white here. So it's white, dominant.
So it's X-linked recessive.
Yep. So white is X-linked recessive. That would be your answer on part A. X-linked recessive. And the easiest way to say that is that the F1 males had one red allele, but they also had one white allele. All of them turned out to have red eyes. That tells you that red is dominant over white. All right, part B. Draw a Punnett square that depicts the cross that produced the F1 flies. Who are the parents of the F1s?
A wide-eyed female, wide-eyed male.
Yeah, the P generation, the original parents. So up here on the original parents, mom up here was X little r, X little r. That's our original mom. And I'm going to have to write this a little bit smaller so I have some space here. And then dad was X big R and Y. So when we cross these guys, this one turns out to be X big R, X little r, X big R, X little r. What does that tell us about every F1 female?
They're all red.
They're all red, right? Because they got the big R. On the other hand, the males here comes out to be X little r from here and Y from here. X little r from here and Y from here. So that tells us that all the F1 males have white eyes. That backs up that statement. Now, on the other hand, out of 100 F2 flies, we get these babies. Well, the F2s are the children of who?
F1.
The F1s. So the next slide is going to ask us to draw a Punnett square to show how that happened. So let's go to the next slide. Draw a Punnett square that depicts the cross that produced the F2 flies described above. So remember, in the F2, we had a female like that, crossed with a male who had white eyes like this. And what I'm hoping that you see is that 1/4 of the babies are going to be what phenotype? What's the phenotype of this box that I filled out?
Red.
Red and?
Red.
What else can we tell about that individual? Red eyes and?
Female.
Female. When we're dealing with sex-linked traits, we need to break it down by trait and sex at the same time. All right, what about that one? What can we tell about that one?
Wide eyes and female.
Wide-eyed female. What about that one?
Red-eyed and male.
Red-eyed and male. And what about this one down at the bottom?
Wide-eyed and male.
Wide-eyed and male. So our Punnett square is telling us that in the F2, we essentially ought to get 1/4 of each possibility, right? 1/4 red-eyed females, 1/4 white-eyed females, 1/4 white-eyed males, and 1/4 red-eyed males. That's what the science is telling us we should get. Now, let's look at question C. Actually, D. Out of 100 F2 offspring, how many were expected to be wild-type males? So if we had 100 F2 babies, how many would we expect to be wild type, which is red eyes, by the way? How many would we expect to be red-eyed males according to this Punnett square?
25.
5.
25, right? 1/4 of the total. 1/4 of 100, 25. We would also expect to get 25 wild type females, 25 white-eyed males, and 25 white-eyed females. Does that make sense? So we're to get our expected when we're dealing with Punnett squares, take the total number of offspring, multiply by the fraction from your Punnett square. That's how you get your expected numbers. Next, we got the null hypothesis. So we're expecting 25 of each. But if we back up a slide, we don't have exactly 25 of each, right? We got 23, 26, 25, 26. In the real world, are you going to get 25, 25, 25, 25 every time? Is that what the Punnett square means?
No.
No. It means on average, that's what you're going to get. You're not going to get that every time you make 100 babies. What we're trying to figure out is, are these numbers close enough to 25, 25, 25, 25 that the differences are probably due to what? Chance. That's really what we're trying to figure out. But our null hypothesis is going to be what?
That there's no statistical difference.
Yeah, there's no statistical difference between our observed numbers, these guys, and our expected numbers, the 25, 25, 25, 25. That's our null hypothesis. So I would just state it, is that there's no statistically significant difference between our observed values and our expected values. That's really all you need to say. Okay, so now we want to calculate the chi-square value. And let's remember our numbers. So actually, let's just go to this table. So we've got a table here. So I'm going to just write it this way. I'm going to write it red-eyed male, red female, white male, white female. So again, we observed 23 wild type females, those are the red eyes, and 26 wild type males. So we had 26 here, 23 here. And then if we go back, we had 25 white-eyed females and 26 white-eyed males. And again, typically what we would do is we would add these numbers up. And if you add those up, you get 100. And then to get those expected values, we multiply by the fractions from our Punnett square. Our F2 Punnett square said we ought to get 1/4 of each of these. 1/4 of each, 1/4 of the total, 1/4 of 100 gives us 25, 25, 25, and 25. And then we fill in our table, just like we did before. So O minus E, 26 minus 25, 1. 23 minus 25, minus 2. 26 minus 25, 25 minus 25. Now this says to square this column. So 1 squared, -2 squared, +4. 1 squared, 0 squared. All right. Then this last one says take this column and divide it by this column. Now you can use a calculator on the AP exam. You can use a 4-function calculator. You can use a scientific calculator. You can use a graphing calculator. You can essentially use any calculator that won't get on the internet. If it'll connect to the internet, you can't use it. But I'm going to tell you, in this case, you probably don't even need a calculator, because I would just do this one in fractions. And what I would do is I would take 1 and divide it by 25, 1/25, like so. Next one would be 4/25. This would be 1/25. And this would be 0/25. And remember that, and you could go ahead and divide these out and put them in decimal if you wanted to. You could go 1 divided by 25 and write 0.0 whatever it comes out to be. That still works. But remember that your chi-squared formula is the summation of O minus E squared over E. So that's essentially, again, telling us to take this last column and add it up, sum it up. So let's do that. And if you did it in fractions, it makes it faster. So 1/25 plus 4/25 plus 1/25 plus 0/25 just comes out to be 6/25. Now I'm going to convert it to a decimal. So I'm just going to go 6 divided by 25. And that gives me 0.24. Now remember, by itself, that doesn't really tell you much. You've got to compare it to that critical value from the table, which is on your formula sheet. So let's go back to that table. Here's our table. How many different phenotypes do we have in that problem?
4.
4, which means how many degrees of freedom are we going to use in this table?
3.
3, always one less than your number of phenotypes. Remember, unless you're told differently, you're always going to use this p-value, which again is sort of an error measurement. We know there's a 5% chance we're going to make a bad conclusion. That's okay. Our critical value, though, is that cell right there, 7.82. So let's go back to our chart. So we had a critical value, or a chi-square value of 0.24. We had a critical value of 7.82. We compare those. In this case, our critical value is greater than chi-square. What do you do when your critical value is greater than your chi-square value? What does that tell you?
You accept the null hypothesis.
Yeah, so they don't like us to say the word accept the null hypothesis. The statisticians want us to say we're going to fail to reject the null hypothesis. What does that mean if we're failing to reject the null hypothesis?
That means the differences are due to chance.
Yeah, that means there really are no statistically significant differences. And that the small differences that did exist were probably just what kind of differences? What caused those differences?
Chance.
Chance. We're just looking at chance, random differences. Nothing important. All right. Questions on this one? All right, I want to work one more quick problem. I know we're running out of time, but I can do this next one really, really fast, probably in 2 or 3 minutes. So here's one other quick problem. So a researcher found that onion root tip cells spent approximately 90% of their life cycle in interphase and 10% in mitosis. Quick reminder, by looking at a cell under a microscope, how can you tell if it's in interphase or if it's in mitosis? Just by looking at a stained cell. What do you see in mitosis that you don't see in interphase?
The
Cells
chromosome.
dividing.
Yeah, so you don't see the chromosomes at all. During interphase, the DNA is unwound and sort of spread out. It's in a form called chromatin. But when mitosis starts, that chromatin winds up around little proteins called histones. It condenses, and you see the chromosomes. So we can tell very quickly by looking at a stained cell whether it's in mitosis or it's not, whether it's in interphase. Interphase is just what's between cell divisions, right? It's when the cell's doing its job, whatever it's supposed to do, but it's not dividing. That's interphase. All right, so the researcher then treated these onion root tips with lectin, a certain chemical. You may not know what that chemical does. You don't have to. And the cells treated with lectin, he found that 700 were in interphase, 1,600 were in mitosis. Stated null hypothesis for this experiment. What's our null hypothesis always going to be? There is no statistically significant difference between the number of cells in interphase and those in mitosis between our experimental group and our theoretical population, or however you want to state that. There's no difference between these numbers and these numbers is essentially what we're saying. So next we want to do a chi-square value. So we got a table just like we had before. And our phenotypes are just going to be, I'm going to abbreviate interphase and mitosis. That's it. And when we actually looked at the cells, we stained them. We had 700 interphase and 1600 in mitosis. So 700 here, 1600 here. How are we going to come up with those expected values? Somebody help me out.
We had 90% expected of in interphase and 10% mitosis, so we're just going to calculate that from.
Yeah. So they're expecting 90% to be in interphase and 10% to be in mitosis. So what we need to do first is add up our total number of cells. 1600 plus 700, get 2300. Multiply 2300 by 90%, 0.9. And that gives us 2070. All right. Multiply 2300 by 10%, which hopefully you don't really need a calculator to do that, to get 230. So just by looking at the data, what can we probably already conclude without even doing the math?
There's a huge difference.
There's a difference, and that difference looks like huge, right? It looks really big. But let's go ahead and do the math. So let's go O minus E. So that's 700 minus 2070. So that comes out to be negative 1370. And then 1600 minus 230. That comes out to be positive 1370. And then in this column, we need to square this column. So 1370 squared. We get this huge number, 18,76900. That's going to be the same right here. And then to get the last column, we need to divide this column by the expected column. So 18,76900 divided by 2070. That gives us 906.71. And by the way, if you're doing chi-square in decimal form, I would always carry it out to at least a hundredths place, just to make sure you don't get rounding errors. Carry it out to at least 2 decimal places. So to get this last one, 1876900 divided by 230. All right, and we get 8160.43. All right, remember our chi-square statistic is the summation of O minus E squared over E. So we need to add up this whole column. So 8160.43 plus 906.71. So we end up with a chi-square value. I'm not sure what's going on with my pen here. Of 9,067.14. We need to compare that to our critical value. So let's go back to our table. Here's our table. How many degrees of freedom in that example problem? Somebody help me out. How many degrees of freedom should we use?
1.
Just 1, right? Because we had 2 phenotypes, interphase or mitosis. Our degrees of freedom were always 1 less than that. We're always going to use that 0.05 probability level. So our critical value is 3.84. Get back to our problem. So we compare these two numbers. In this case, chi-square is much greater than our critical value. And when that happens, what does that tell us?
There's a statistical difference.
Yeah, we're going to reject the null hypothesis. And that tells us we're rejecting the idea that there are no differences, which means what?
But there is a difference.
There are differences. And what is this telling us this lectin chemical is doing?
Changing the time that they're in interphase and mitosis.
Yeah, so it seems to be causing mitosis to happen, right? It seems to be stimulating mitosis because we expected only 230 cells in mitosis, but when it was treated with lectin, instead we had 1,600. So it's a stimulator for mitosis. It's causing mitosis to happen faster than it would normally happen. All right, I had a couple of other things that I wanted to do today, but we're already about 10 minutes over. So I'm going to call it there, but I'll hang on for questions. And then what I had planned to do later on, I'll put that into next week's session, and we'll go from there. So next week may be a continuation of Unit 1, because there's a couple more Unit 1 things that we needed to cover that we didn't get to today. Can I answer questions for any of you guys? All right. Well, thank you guys for giving up an hour and 15 minutes of your afternoon for being here. I appreciate it. And like I said, we're going to do these every Monday, same time, same link. And hopefully by next week, we'll have the Pear Deck thing worked out so it'll be a little bit more interactive and you can input some answers as you go. But again, thanks for being here. I appreciate it. I hope it was helpful.